There's a newly discovered gene editing system, and it might be better for gene therapies than anything we currently have. This is another paper from Feng Zhang's lab, which is kind of on a roll right now. Anyway, to put this paper into context, let's talk about CRISPR for a second. So in case you haven't heard, CRISPR-Cas9 is a gene editing system now commonly used in research and in some therapeutics. The system is derived from bacteria, which use it as a defense against invading viruses. It takes DNA from a virus's genome and then uses that to recognize invading viruses' DNA so that it can be cut up. But here's a question. Where did Cas9 come from? Because proteins evolve over time with organisms, so surely there are earlier versions of Cas9 in the evolutionary record, right? And if you could find those earlier versions of Cas9, maybe they would also be useful DNA cutters. Well, another group had previously postulated that Cas9 was derived from this protein called ISCB because it has a similar sequence. But no one really understood what ISCB does or how it evolved into this programmable CRISPR system where the DNA cutting enzyme is guided to a target site by an RNA. So to try to answer these questions, the researchers looked at all the ISCB genes across the bacterial evolutionary tree, and to summarize a lot, they found that many ISCB genes are associated with an RNA. They call this associated RNA an omega RNA, and omega RNAs were often found right next to the ISCB gene. And this is what it looks like. It has all these loops that can help RNAs bind to specific proteins, and it also had this one sequence that tended to be pretty different across ISCB genes. And so this part was thought to be a guide that could lead the ISCB and omega RNA pair to a DNA target to cut. So they confirmed that this ISCB system is capable of cutting DNA at sequences specified by the omega RNA, meaning it's functionally similar to CRISPR. Here we see an ISCB system successfully producing edits in several target genes in human cells, albeit at pretty low efficiencies. So given the low efficiency, why is this exciting? Well, ISCB is much smaller than Cas9. It's about 30% the size. That's a really useful quality for genome editing in organisms because it's generally easier to get smaller cargoes into cells. And the lower editing efficiency isn't necessarily a cause for concern because these editing systems can be engineered to have greater efficiency. This was done with CRISPR-Cas9 and its derivative systems, and the authors of this paper suggest that it can be done again for ISCBs. So anyway, I'm really excited to see how ISCBs are further developed for editing in human cells and whether they can improve gene therapies.